PhD at Davis with Mike Torelli, who was a sabbatical visitor here for a while last year. Lindy's got sort of two threads to her dissertation work. One's on ecological genomics in Melanogaster and its relevance. The other part is the subject of the talk today, connecting host adaptation and reproductive isolation in Euphydryas editha, or editha, I'm not sure how to pronounce it. Lindy is around this afternoon as well, for those who want to chat with her. So welcome. Thanks for coming on.
Thanks. Really appreciate the opportunity to talk today. Michael, one of my advisors, who you probably know, was here a year or two ago, really raved about his interactions here at the museum, and I actually came down to hear Harry Green's talk last week, and I was really impressed not only by the talk but also by the number of people who came out and the atmosphere and interaction in the room. So thanks for the opportunity, and please interrupt me at any time if you have questions or comments. The work I'll talk about is the product of a collaboration between myself and Mike Singer at the University of Texas. And the question that we're most interested in here is, what is the role of ecological adaptation in speciation? This is a really big question, and it's an old question. And I think most people would agree that ecological adaptation does play some role in speciation. But exactly what role it plays is not well understood. One possibility involves this phenomenon called extrinsic postzygotic isolation. This occurs when hybrids between ecologically divergent populations are unfit because they have intermediate phenotypes that fare poorly in the niches of their parents. So let me explain this with an example. Around 10,000 years ago, in a few lakes in British Columbia, tree-spine sticklebacks began adapting to use different resources. The Linnetic morph here has a slender body and a small mouth, and it's adapted to feed on zooplankton in the water column. The benthic form in the back has a larger body and a wider mouth, and it's adapted to feed on small invertebrates in the shallow margins of the lake. So we have divergence in two ecological traits, body depth and mouth gait, that lead to these two forms sitting on two adaptive peaks, corresponding to two different types of resources. Hybrids between these forms are intermediate, and they fall into this adaptive valley. Hatfield and Schluter in 1999 showed that they grow slowly in both Linnetic and benthic lake enclosures, despite the fact that they actually do just fine in a neutral laboratory setting. So they argued that the problems hybrids faced in the lake enclosures are due to a match between the hybrid, or a mismatch rather, between the hybrid and its environment, and not to inherent developmental defects, since the hybrids did just fine in the laboratory. So again, this is called extrinsic postzygotic isolation. It's isolation because it represents a barrier to gene flow. It's postzygotic because the barrier kicks in only after hybrid zygotes have formed, and it's extrinsic because reduced fitness in hybrids results from a mismatch between the hybrid and its external environment, rather than some inherent defect. So why should we be interested in this? First of all, unlike other forms of reproductive isolation, it's a unique prediction of ecological speciation. So its existence in and of itself provides evidence that ecology can drive speciation. Secondly, it's potentially important during the early stages of speciation, and therefore can possibly trigger processes that kick in later, like reinforcement. And lastly, we have very few data available on the strength and prevalence of this phenomenon in nature. So I did a quick literature search, and I gathered all the studies I could find that provided a quantitative estimate of the strength of this type of phenomenon among natural populations. And you can see I only found about 12 studies, and only 2 of these 12 explicitly tested the idea that reduced hybrid fitness was extrinsic in nature rather than intrinsic. So a third thing I want to point out is that almost all of these studies address sympatric or parapatric populations. These are populations that have proven their ability to coexist despite gene flow or potential gene flow in secondary contact, and are arguably well beyond the early stages of speciation. So what we have here is few data in general, even fewer data at what I would say is probably often the first stage of speciation, ecological divergence in allopatry. So I'm going to modify the question here and focus on whether extrinsic postzygotic isolation is important at the early stages of ecological divergence and allopatry for the rest of my talk. Mike and I adjust this question by examining hybrids between allopatric populations of a single butterfly species that are adapting to use different host plants. In the butterfly, as you already know from my title, is Edith's checker spot, or Euphydryas editha. This is an invalid, and it occurs in local scattered populations across western North America, from southern Canada down to northern Baja. In the populations that we study, it completes just one generation per year. So adults eclose in the early summer. Females usually mate within a day of eclosion, and they mate just once. They lay their eggs in clusters, and after 1 to 2 weeks, those clusters hatch into groups of gregarious young larvae. Larvae will feed for about 3 instars and then enter diapause, where they'll stay all winter, often buried under several feet of snow, until the next spring, when they resume feeding, become active again. At this point, they're solitary and they wander around feeding opportunistically. So in the late summer, we have young larvae that are gregarious and confined to the host that their mother put them on. And in the early spring, we have older larvae that are solitary and wandering around, making their own decisions about what to eat. These older larvae feed for about 2 to 3 weeks before they eventually pupate in the litter on the ground. Pupae develop for 1 to 2 weeks and then emerge as adults in the summer. So the most host-specific stages of this life cycle are the adult females and the young larvae. The adult females are specifically adapted to find and lay their eggs on particular host plants, and the young larvae are specifically adapted to survive on and digest those host plants. The host for most populations of Euphydryas editha is Indian paintbrush or castor. Within California and Oregon, however, butterflies use plants in any of 5 different host genera, including Caspilea, Pedicularis, Penstemon, Plantago, and Colinzia. This doesn't mean that butterflies in California are less host-specific than butterflies outside of California. In fact, even within California, local populations are highly specialized. So this shows data from about 35 populations in California. And each pie represents the proportion of eggs laid on plants in the different host genera. And the first thing to notice is that most of these pies have just 1 or 2 colors, which means that most populations specialize on just 1 or 2 genera. Second thing to notice is that different populations often specialize on different host plants. I'll be focusing in particular on populations that use either Pedicularis or Colinzia. And in this case, the difference in host use between populations is not a simple result of differences in host availability. So at most of these sites, both hosts occur, but butterflies choose to lay their eggs on one and ignore the other. So the differences are caused by genetically based differences in female host preference. The second thing I'd like to point out about these 2 types of populations in particular is that they're not geographically differentiated. So they both occur in the coastal range, they both occur in the Sierra, they both occur in the north, and they both occur in the south. So these are not geographic subspecies. These populations are also not differentiated by physical habitat. This is a photo of our main study site, Sequoia National Forest. Local butterflies here are adapted to Pedicularis. And on the right is a photo from another one of our study sites near Tahoe, about 100 or 150 kilometers to the north, and local butterflies there are adapted to use Colinzia. So you can see that the habitats look very similar. Finally, 2 studies from Mike's lab showed that populations on Colinzia and Pedicularis do not show consistent genetic differentiation. On the left, we have a multi-dimensional scaling plot that summarizes genetic variation at 200 aphlops. So each dot on the plot represents a single population, and the further 2 dots are from each other on the plot, the less similar the 2 populations are genetically. You can see that the blue dots and yellow dots here do not form 2 distinct clusters. On the right, we have a parsimony tree based on restriction enzyme digestions of mitochondrial DNA. And again, you can see that blue and yellow haplotypes don't seem to form distinct clades. And in particular, a few haplotypes are shared. Those haplotypes that have both blue and yellow dots are shared between the 2 types of populations. So what is different about these populations? Well, they use different plants. So here's a sketch of Pedicularis. This is a large herb. It has a basal rosette. It's relatively difficult for caterpillars to digest. It's a perennial that retains water throughout the summer, and it receives it's heavily grazed, and it's frequented by predators, by insect predators. Colinzia, on the other hand, is quite different. It's a small, dainty, upright plant. It's highly nutritious. It supports rapid growth of caterpillars. It's an annual that senesces quickly, and it receives little grazing and is not visited very often by insect predators. clusters. Larvae adapted to Pedicularis can digest both Pedicularis, obviously, and also Collinsia, whereas larvae adapted to Collinsia cannot digest Pedicularis. They simply don't survive. Mike and I discovered a few summers ago that larvae adapted to Pedicularis tend to crawl down. They're positively geotaxic, and they feed and rest at the base of their host plant. While larvae adapted to Collinsia are negatively geotaxic, they tend to crawl up, and they feed and rest at the top of their host plant. Adult females adapted to Pedicularis lay large clusters of 30 to 60 eggs, and those adapted to Collinsia lay smaller clusters of 10 eggs. In Pedicularis, females, just like larvae, are positively geotaxic, and they lay their eggs at the base of the plant. And on Collinsia, females tend to lay their eggs at the top of the plant. So here we have these two types of populations that are interspersed geographically, that share similar habitats, that don't seem to be consistently differentiated genetically, but that exhibit quite an impressive array of divergent host adaptations. So in light of these host adaptations, I'll ask two questions. First, are hybrids intermediate, and are hybrids less fit? If the answer to these questions is yes, then we will have shown extrinsic host-dynamic isolation in this system. I'll focus on the life stages that are most host-specific. In particular, adult female host preference, geotaxis, and clutch size. Mike and I have also looked at larval digestion and larval geotaxis, and I'll mention those results right at the end, but I won't have time to go into them in much detail. So to start with, all the hybrids we used in our experiments came from the following series of crosses. So this is again that plot showing nuclear genetic variation at 200 eclipse. We chose two Collinsia adapted populations, they're in blue, and two Pedicularis adapted populations in yellow. From one of the Collinsia adapted populations, we took virgin females and mated them to males from the other Collinsia adapted population, producing offspring we refer to as CC. Both parents adapted to Collinsia. With another group of virgin females, we mated them to males from the two Pedicularis populations, producing offspring we refer to as CP. We did the same thing with virgin females from one of the two Pedicularis populations, producing PP and PC offspring. Notice that these two populations, for which we gathered all the females for our crosses, are more closely related to each other, both genetically and geographically, than they are to the other populations. So we should be able to isolate the effects of host adaptation separately from genetic or geographic distance. And I'm going to go over this just one more time because I'll continue to use these abbreviations and phrases for the rest of the talk. We have CC individuals, I'll call them fully adapted to Collinsia, and represent them with blue because Collinsia has blue flowers. Fully adapted Pedic individuals, PP, I'll represent them with yellow because Pedicularis has yellow flowers. And then we have these hybrids in the middle, two reciprocal types. The first letter corresponds to the host relation of the mother, the second to the host relation of the father, and I'll represent them with green. We chose to compare the hybrids to these fully adapted individuals, whose parents also come from allopatral populations, so that we could examine the effects of host adaptation while controlling for genetic drift in allopatry. It turns out in the end that these so-called fully adapted individuals are phenotypically indistinguishable from the pure types, pure offspring within population crosses. So in the end, it doesn't actually matter much. All right, so first host preference. Are hybrids intermediate in host preference, and are they less fit? Typically, what entomologists assay host preference, they put the insect in a cage with the two plants, and they come back a few hours or a day later and count the number of eggs that were laid on each host. This doesn't work at all for our insects. They simply won't perform a natural search behavior in a cage, and what they do is they fly to the screen and they sit there for the rest of the day. Exactly. They may eventually become agitated enough to fly around and run into one of the hosts by accident, but all the noise, I mean, it's really haphazard and it creates a lot of noise in the data. So it's kind of like putting a blindfolded man in a room with a carrot and a piece of cake and coming back the next day to see how much he's eaten of each item. There's going to be a lot of noise associated with what he happened to run into first and how hungry he was when he ran into it. A better option for testing this man's preference might be to offer him both items directly and simultaneously in a choice test. But this also has its problems. First of all, it at best provides qualitative information. He prefers the carrot or if he prefers the cake. And at worst, it can provide misleading information. If he's not hungry enough, he may not accept either item. And if he's too hungry, he may accept both without expressing a preference. So Mike and I would suggest that a better paradigm for testing the food preference of this individual would be to conduct a sequential no-choice test. The idea is that you start when the person's not hungry enough to accept either item. So you offer them the cake, he rejects it. You offer him the carrot, he rejects it. Eventually, he may become hungry enough to accept the cake, but you can't let him eat it. First of all, you don't yet know whether he prefers the cake to the carrot, because it's possible that if you'd offered him the carrot this time, he would have accepted that, and he's just all of a sudden become hungry enough to want food. So you have to take the cake away and ensure that hunger increases over the course of the test. So you offer the carrot again, he still rejects, he accepts the cake, he rejects the carrot, he accepts the cake. At this point, it's clear that he does prefer the cake rather to the carrot. And another thing to point out is that once a food item is accepted, it's always accepted every time it's offered thereafter, because hunger is increasing. So if he was hungry enough to accept it here, he should be hungry enough to accept it every subsequent time. So the test continues, reject the carrot, accept the cake. Eventually, he may accept the carrot. And this provides a quantitative estimate of the strength of his preference for the item he likes over the item he doesn't like. Namely, the length of time it takes him to accept the item he doesn't like after having accepted the item he does like. Mike's called this the discrimination phase because it's the period of time over which he's discriminated between the two items. We basically do this exact same thing with our butterflies, except instead of offering them food, we offer them host plants and ask whether they want to lay their eggs on them. You couldn't do this with all insects because most insects can't be handled so easily, but it turns out that these butterflies and other butterflies in the same subfamily are extremely docile. So you can pick up a butterfly, as you see here, put her onto a host plant, and she acts as if she just landed on the host plant herself. And she immediately starts tasting it. If she likes it, she'll look around for a spot to lay eggs and curl her ovipositor. Once she's fully curled and her ovipositor is exerted, it's clear that she wants to lay eggs and that she will lay eggs. And so at that point, you can pull her off before the first egg actually comes out. So this is where all this happens in a preference tent. Pedicularis is a parasitic plant, so you can't pot it. Its roots actually tap into the roots of conifers. So you take a tent out in the field, cut a hole in the bottom, stick it over a naturally growing pedicularis plant, and then you bring Colinzia in pots. And one at a time, you take butterflies out of their cages, and you ask them whether they want this, whether they want that, whether they want this, whether they want that. Here's a typical discrimination phase for a pedicadaptive female. Once she's accepted pedicularis, she usually waits about 4 hours, sometimes up to a day, to accept Colinzia. Colinzia-adapted butterflies have much stronger preferences. They'll often wait 2 to 3 days after having accepted Colinzia to accept pedicularis. Sometimes they'll die without ever having accepted pedicularis. And this makes sense because their larvae can't survive on pedicularis. So now I'll show you some real data. On the y-axis, we have discrimination phase in days. I've arbitrarily used the minus sign to indicate a pedicularis preference, and positive values indicate what the Colinzia preference is. 0 here means no preference. The fully adapted individuals in our tests look just like the pure individuals in the wild. CC butterflies have very strong preferences for Colinzia. They waited at least a day or more before accepting pedicularis. And fully adapted pedicularis individuals had slightly weaker preferences for pedicularis. Sometimes they'd accept Colinzia within a couple hours, and sometimes they would wait a couple days. It turns out that hybrids are indeed intermediate. In general, they don't have strong preferences. There's a little bit of variation up here in the PC type, with a few individuals having moderate to strong preferences for Colinzia. And this may result from sex linkage. In Lepidoptera, the females are heterogametic. So this female has inherited her Z chromosome from her Colinzia-adapted father. So if preference loci reside on the Z chromosome, then we would expect PC individuals to resemble CC more than the CP individuals do. Okay, so host preference is intermediate. This intermediate host preference incurs some sort of fitness cost. It's hard to see how an intermediate host preference in and of itself would be necessarily bad. But I'll come back to this later because it's quite possible that intermediate host preferences interact with other phenotypes to reduce fitness. For now, I'll move on to geotaxis. And I'll ask, are hybrids intermediate in geotaxis? And is intermediate geotaxis, does it confer the fitness cost? Before I get to the hybrids, I'm going to show you two videos illustrating the difference between the pure types. So the first video shows a Colinzia-adapted individual laying on Colinzia, and the second shows a Vertebrate-adapted individual laying at the base of a Vertebrate plant. It's a little bit hard to see, but this is a small Colinzia plant in the middle of the video there that's tilting away from the camera. Here comes the butterfly. You can see how docile they are. She'll just walk onto the plant. She's tapping it with her foretarsi. That's what she's doing there in front. And she's already fully curled. This is her abdomen here, curled under the leaf, looking for a spot to lay. It's hard to see, but she's actually completely suspended in air. Her wings aren't touching the ground. And I'm fast forwarding here to the point where she lays her first egg. And there it is. So it's not exactly true that these butterflies have a really strong instinct to lay at the top of the plant. They just don't seem to be very picky. If they accept the plant, they'll lay wherever they happen to be, wherever they land or wherever the experimenter places them. The next video shows a Vertebrate adapted female. Her instincts, on the other hand, are very strong. So here I'm nudging her to get her to wake up and remember that she's on the plant. Okay, she's dipping her antennae, so she's interested, she's smelling the plant. And instead of being content to lay where she is, she immediately drops down to the lower leaf and eventually makes it all the way down to the ground. This plant's a little bit spindly, so there doesn't happen to be any host material on the ground there, so she spends some time looking around and then flies back to the plant. Again, she's excited, but she wants to lay on the ground, so she heads back down. This time she does end up actually flying to and finding the base of the plant, and now she's just looking around for a place to lay. So you can see that her instincts are really strong, and the behavior is actually really discretely different from the behavior of the Colinzia-adapted females. Here I'm going to zoom in and show you the spot where she actually decided to lay. This is the dried up flower capsule on the plant. It's about a centimeter off the ground. You can see her abdomen going in and out of the flower. Every time it goes in, she's putting another egg in her clutch. So how about hybrids? Here I'll show you a hybrid being offered the exact same Euphydryas plant we saw in the last video. This is hard to see because she's in the background, but she's actually already fully curled and sitting there ready to lay her eggs. When we first saw this, it was just so striking to us because you just never see this behavior in nature. A Vertebrate-adapted female would never lay this high off the ground, and a Colinzia-adapted female would never accept this plant so readily. And there's her first egg. So to quantify these differences, every time one of our females accepted Vertebrate during our tests, we recorded the spot where she decided to lay. We always placed the butterflies at least 5 centimeters per hybrid. So if she ended up laying up here, it means probably she just laid wherever she was placed. And if she ends up laying down here in the gray area, it means she must have dropped down from the place where we put her. And here you can see the fully adapted types. Not very many Colinzia-adapted females accept Vertebrate, so there's very few data here. But in general, they lay where they were placed. And the Vertebrate-adapted individuals always drop down. Hybrids, again, are intermediate. There's a lot of variance here. Like you saw in the video, most hybrids are willing to lay wherever they are. But we did see a significant number of hybrids that exhibited geotaxic behavior. And in watching them on these plants, it really did seem like their instincts to go down were just as strong as some of the Vertebrate-adapted females. So there's almost a discrete difference here segregating between hybrids. In order to look at to estimate the fitness consequences of not being geotaxis, right, laying at the top, we place neonates out in the field, one group on a high leaf and one group on a low leaf on each of 10 plants. So the experimental unit here is the plant. We left the caterpillars there for 10 days. We came back, collected them, weighed them, and counted them. And you can see there was a striking difference in growth rate. So this on the y-axis is residual growth rate after having controlled for the plant effect. In every single case, larvae in the low clutch grew faster than larvae in the high clutch. We think this is probably because there's this buffer of really hot air near the ground, since the ground is radiating heat. And so they end up just existing at a higher temperature in the low clutches than they are in the high clutches, and developing more quickly. We saw no such difference in survival, but we do actually think that laying high in pedicularis may have survival consequences, and that's because this plant is heavily grazed. So you can imagine that if you lay your clutch right here, you're much more likely to be accidentally taken by a predator by the time your eggs hatch than you are if you laid right at the base, like we saw that for Lindy in the video. How about geotaxic phenotypes and fitness on Collinsia? Collinsia doesn't really offer any places to lay near the ground. And in fact, pedicularis adapted females, these positively geotaxic insects that accept Collinsia, often have a hard time laying because they accept the plant, they dump it around, and they can't find it, and they eventually give up. And those few hybrids we had that seemed to be positively geotaxic, they face the same problem. So here's another video. This is a positively geotaxic hybrid being offered Collinsia. She's tapping and curling. But she's not happy to lay way up there, and she goes down to the ground, and eventually she gives up. So in summary, not being geotaxic, most hybrids are not geotaxic, and their offspring are expected to grow more slowly on pedicularis. A few hybrids are geotaxic, and they're expected to have problems laying on Collinsia. Sometimes they may simply fail to lay. Now I'll talk about clutch size. These two photos illustrate the difference between the peer types that I already mentioned. Here's a single egg that was laid on Collinsia by a Collinsia adapted female. And on the right, here's a pedicularis adapted female laying a large cluster of eggs on pedicularis. The fully adapted insects in our tests replicated these differences. So we have CC individuals laying an average of about 2.5 eggs on Collinsia. And we have PP individuals laying an average of about 50 eggs on pedicularis. You notice that these two types of insects laid slightly more eggs on the host that they didn't like than they did on the host that they did like. So CC laying about 20 eggs on Pedic and only about 2 eggs on Collinsia. That's because these individuals have been waiting for days to lay eggs because they really don't like the plant. And by the time they finally accept it, they're highly, highly motivated, almost desperate to lay eggs. So they end up laying more eggs than they otherwise would. Hybrids, we found, lay, again, intermediate-sized clutches of 15 to 30 eggs on both hosts. And one way to think about this is, how many eggs do butterflies lay on the host that they prefer, their intrinsic clutch size? You can see that on Collinsia, hybrids lay larger clutches than Collinsia adapted butterflies. And on Pedic, they lay smaller clutches than Pedic adapted butterflies. To examine the fitness consequences of this behavior, we compared the growth and survival of small and medium-sized clutches on Collinsia and medium and large-sized clutches on pedicularis. And we did this by putting groups of larvae out in the field in different numbers. So on pedicularis, we put groups of 5, 10, 20, and 40 larvae, each clutch on a different plant. We replicated this block structure 18 times. Again, we left them there for 10 days, exposed to predators and the elements, and we came back and collected them. It's useful if these larvae don't move around, because it means you can do this kind of thing without putting them in enclosures. Things like that. So we didn't find any differences in growth rate associated with clutch size, but we did see significant differences in survival, and you can see in general it looks like survival increases with clutch size. On Collinsia, we put out smaller clutches, 1, 5, 10, 15, 25 larvae, and the opposite was true. Survival seemed to decrease with clutch size. So to visualize this, I've just fit splines to the mean survival estimates for each clutch size. You can see this increasing trend of pedicularis and this decreasing trend on Collinsia. We don't fully understand what the mechanism is here on the right, but at least on pedicularis, this is mediated by predators. Because if you exclude predators, the effect completely goes away. So somehow caterpillars are benefited from being in groups. They defend themselves better that way. So I mentioned earlier that, well, so far I've just shown you the fitness effects of all these phenotypes in isolation. Eventually it'd be nice to combine different phenotypes and ask, for example, what's the consequence of an intermediate host preference combined with a small clutch size, or with an intermediate clutch size, or with a large clutch size? And to do that, I'm going to return to the adaptive surface type of figure that I showed you at the beginning of the talk. In this box, I'll draw a surface whose height here on the z-axis corresponds to the expected survival of offspring of females with particular host preference over there, that clutch size here, phenotype combinations. So here's the surface. You can see in the foreground, females have long discrimination phases for Collinsia. So for these females, they'll be laying on Collinsia in nature, and small clutch sizes have higher survival than large clutch sizes. In the background, you have females who are waiting at least half a day or more to accept Collinsia after having accepted Pedicularis. So these females prefer Pedicularis and will lay on Pedicularis in nature. For them, small clutch sizes have less survival or lower survival than large clutch sizes. And then in the midground here, you have this rapid transition in host preference. As the discrimination phase goes from -1/2 a day to +1/2 a day, butterflies go from almost always laying on Pedicularis to almost always laying on Collinsia, and the survival of a large clutch goes from being relatively high to relatively low. So all this data is based on the information I just gave you. This curve here is exactly the same curve that I fit to the data relating clutch size to survival on Collinsia. And this curve in the back there is exactly the same curve that I fit to the Pedicularis survival data. Then, using the phenotype data that we collected, I simulated the position of females of different types on the surface. So each dot represents the one hypothetical female, and there's a lot of scatter here associated with the noise and uncertainty of the data. But in general, the two full adapted types are sitting on two adaptive peaks, corresponding to small clutch sizes on Collinsia and large clutch sizes on Pedicularis. And the hybrids are right in the middle in this adaptive valley. They have intermediate host preferences and intermediate clutch sizes. So this, I think, sort of allows us to visualize what's going on here. This is extrinsic post-zygotic isolation, and it's surprisingly strong. And it indicates that this type of phenomenon can be strong among closely related populations that are adapting to different ecological niches. And remember, this doesn't yet incorporate the reduction in fitness that I talked about associated with geotaxis, or that's associated with larval phenotypes. So here's a table summarizing the strength and source of hybrid maladaptation. And you can see that intermediate clutch size leads to 29 and 42% survival deficit on Pedicularis and Collinsia respectively. That's what you saw in the last adaptive surface plot. Intermediate geotaxis leads to about a 42% reduction in growth on Pedicularis and can lead to failure to lay on Collinsia. And then I didn't show you these data, but we found that hybrids do not digest Pedicularis as well as Pedicularis adapted larvae do. Their growth rate is reduced by about 23%. And larval geotaxis on Collinsia leads to reduced growth because when the larvae feed at the bottom, they end up feeding on these less nutritious basal leaves, and the larvae that feed at the top end up feeding on these highly nutritious young leaves and buds. So after having collected all this data, the question that I couldn't help but ask is, why the hell have these populations become different species? I mean, they're so different that morphologically you can't tell them apart, but if you go out into the field and pick up a female, there's no question who she is or what she's adapted to. And it's just remarkable that you can have such striking differences within a single species. So how could this be possible? Well, one thing which I haven't mentioned yet and which I've actually actively denied is that maybe there already are different species. It's theoretically possible that these things would never agree to mate with each other in nature. I think it's really unlikely because we had no problem getting them to mate with each other in small cages, but we haven't actually done the field tests that would be required to determine whether that reflects natural behavior. Another possibility is that they will eventually become new species, and this is kind of what I was hoping when I started this whole project, is that we could catch the speciation process right at its earliest stages. It's hard to know if that's true. but that's sort of the line you walk when you want to study the early stages, and it's so early you don't yet know what's going to happen. Thirdly, it's possible that there's too little migration between populations to trigger reinforcement. These butterflies are fairly sedentary, and if migration is low, hybrids may not be forming often enough to create selection pressure for pre-mating isolation to evolve. So maybe these populations are in this sort of holding pattern, and eventually if migration increases, reinforcement will kick in and they will become different species. And lastly, it's possible that extrinsic post-zygotic isolation alone simply isn't enough. Most of the stories that you're probably familiar with where people have conclusively linked host adaptation to speciation in insects involve organisms that mate on their host plants. And a divergence in host preference itself causes insects not to mate with each other. So it's sort of a magic trait. Our butterflies don't mate on their hosts, and their hosts are available at the same time of year. So, if our hosts were available at different times of year, that would also be a sort of magic trait. If you enclose with this host, you can't mate with insects on that host because you're not around at the same time. So that's another possibility that would be interesting to look into, although difficult to test. Okay, so I want to acknowledge my collaborator, Mike Singer. He's been working with these butterflies for over 20 years, and although he wasn't particularly interested in speciation before our collaboration, he is responsible for accumulating almost all of the natural history data and ecological data on host use and host evolution in the system. He's also developed a lot of the techniques we use to rear and test insects. I got a lot of important advice from my two advisors at UC Davis, Michael Trelli and Sergei Nushin, also from Art Shapiro, Sharon Strauss, and Lawrence Stanley. I had some great field assistants over the 3 summers that it took to conduct all this work, and funding from NSF and UC Davis Center for Population Knowledge. Thank you.
Good job.
Okay, so Lindy's left us plenty of time for questions. Yes, Chris.
So Mike had some long story about rabbit meadow and host shifts. How do they, and I don't remember the actual how it goes, how do they actually shift hosts in the wild after a population from one to the other?
Yeah, so that was a Pedicularis adapted population that shifted to Collinsia. What happened was there was the Forest Service used this sort of slash and burn policy in the 70s. And so there were these big clearings where the Pedicularis died because all the conifers died, and Pedicularis is parasitic on conifers. And the Collinsia was fertilized by the burns. So all of a sudden, the Collinsia became extremely abundant and extremely nutritious. And these butterflies were falling all over it. They had no idea how to use it. But it didn't matter because the Collinsia was so good. And so they actually, for a short period of time, shifted onto Collinsia. Until a frost, one winter, killed all the individuals in the clearings where the Collinsia was growing. And after that, they never went back to it. And at this point, the Collinsia is back to its original state, which is not very nutritious and not usable for an insect that isn't adapted to it. Yeah.
The butterflies that lay eggs on the Collinsia lay fewer eggs. Are they larger eggs, or are they different sized eggs?
Yeah. Thanks. I meant to mention that. They're about the same size, but they lay more often. So both types of females lay about the same number of eggs per day. Females lay many small clutches or one large clutch.
And then just as a follow-up, wouldn't you consider the ability to interbreed a shared primitive character and therefore not necessarily indicative of speciation? Because, two things that can interbreed don't necessarily, that could be something shared by a more distant common ancestor. Well, Galapagos tortoises are separate species and they can interbreed.
Okay.
Yeah, and
do they interbreed in nature?
Well, they do when given the opportunity, and they do on Isabella, which is an island that, for the most part, they are separate populations, but on occasion they interbreed, and there are natural cases of tortoises, going from one place to another and interbreeding.
Yeah.
But they're still recognized as separate taxa.
Yeah. So the species concept I'm using here is the biological species concept, and so I'm thinking to myself, well, if they come into contact, will they breed? Will they collapse into one species, or will they remain two? And if they won't mate with each other, then they're unlikely to collapse back into one. Does that answer your question?
Sort of, yeah.
Does it just betray my biases?
That's fine.
So do you ever see hybrids in the wild? I mean, I know you said it's hard to tell based on overall morphology, but you thought about using microsats or something to see how common it is to get hybrids?
Yeah, we haven't knowingly seen a hybrid in nature, and it would be very obvious. If we saw it, it would be obvious. But if we actually worked with butterfly and tested our preference and our clutch size, it would be obvious. And Mike, in particular, has tested a lot of butterflies and not seen these types of intermediate phenotypes in the wild. As far as microsatellites go, that'd definitely be something to do. It's possible we could find genetic differences in microsatellite loci, and even diagnostic differences that we could use to diagnose hybrids, but we haven't done that yet.
Across all of the different populations, do you see the same sets of adaptations to different host plants in all populations?
Including the populations on the other host genera?
Yeah, well, since you have all your populations adapted to one host and all the populations adapted to the other, some were phonotaxic in a certain way, or they just kind of small clutch sizes. Do those characters always correlate across all the populations for each host plant?
Yeah, so all pedicularis populations lay large clutches at the base of their host, and obviously have the ability to digest the plant. Collinsia, I didn't have time to really go into the details, but the genus Collinsia is not as monomorphic in respect to the butterflies. So the Collinsia populations we worked with was Collinsia torii, which is a small little upright thing I showed you. All populations on that species have these small clutches that are negatively eutaxic. But there are other types of Collinsia that are used in the Great Basin on the east side of the Sierra, for example, that are much larger. And they're using Collinsia, but they actually lay larger clutches. And they do put those at the base of the plant because those large Collinsias have places at the base.
It's very interesting, strong ecological adaptation, repeated so many times in the same
Yeah.
sets of traits. It really is, as you say, not a single genetic relative, right?
Yeah, I didn't mean to imply that I don't think it's a single genetic origin, but I agree it'd be really interesting to look at because you do have this geographical interdispersed populations, and if we could find the genes underlying these behaviors, we might be able to figure out whether it's single origin or not. Yeah.
Do you know how much spatial structuring there is between these populations? I mean, in terms of genetics, is there geographic structure?
Yes, there's a little bit of geographic structure. There's a signal in that aphlet study. Brian Wee, who was one of Mike Singer's PhD students from a few years ago, he did find a signal associated with Sierra versus Coastal Range. And on a very small scale, he found a signal associated with geographic distance within metapopulations in Yosemite National Park and in Sequoia Kings Canyon area.
So you'll get these two host plant specialists in a particular area being more closely related than in neighboring populations of either one of them.
It seems like it. There was a slight signal of host use in the nuclear data associated with Collinsia compared to some of the other hosts, but not compared to pedicularis. So pedicularis popped up kind of right in the middle of the Collinsia cluster.
Yeah, sure.
I'm wondering if you show the plots where you have some populations that have multiple host preferences. And I'm wondering if there's a relationship between the patch size of that resource, the host resource, that varies with the frequency of that host use.
Yeah.
It's a frequency dependent trait, so just you need a patch where the resource is readily available for that host shifting.
I think I understand your question. So the preferences themselves aren't frequency dependent. Like a female has the same preference, which may be expressed differently because plants are differently abundant. So a female always prefers, Pedicta Collinsia, but because Collinsia is more abundant, she actually ends up laying Collinsia more often than Pedicta. And that's true that some of the differences in that map I showed you have to do with host availability and abundance, but not the ones between Pedicta and Collinsia. But you saw that there's this group of coastal populations here, which some of you might be familiar with in the Bay Area, that feed on Plantago. And none of the populations in the Sierra feed on Plantago, and that's because there is no Plantago in our Sierra.
Well, that's a good reason.
Yeah.
Carol.
Where are the males when all this is happening? Like, do they have a preference at all,
Yeah.
or once they pupate, they just don't care what plants they hang around?
Yeah, we can't. No, we don't see any particular association between males and host plants. And males are closed about a week earlier than females, and they just fly around looking for females, and that's basically what they do. And the plants like herd together in the same patches, so they're flying over, looking for females forever. Yeah. And females are closed wherever because the post-diapause caterpillars wander away from the host before they can.
So what about doing the same experiments for male-female preference to determine how likely hybrids are?
Mate preference?
Mate preference.
With the sequential no-choice test?
Why not?
Yeah, that'd be interesting. Yeah, we could do that. The cool thing about this butterfly that allows us to do that preference test is that they act so naturally on their host, despite being handled. And the same may not be true of the mating behavior. So, like in a cage, these two things mate just fine, no problem. Put them in a cage together, you come back an hour later, they're mated. But the female doesn't really have an opportunity to fly away in the cage, and the males are pretty persistent.
I was intrigued by the comment you made that the behaviour of the butterflies with respect to the host plant preference might vary depending on sort of recent environmental conditions, and that sort of brings sort of what the grants have done on the balance of interest to mind, to really understand why things are or not speciating. You've got to look at those sort of dynamics over time.
Yeah.
Do you think that's a really big factor in the system?
I do, yeah, and I didn't talk about that at all. And not even with that host shift situation that Mike has studied in the past, but simply with these populations, a lot of the fitness effects we see on Collinsia are dependent on the year, because the big challenge to insects that use Collinsia is that it's an annual node and senesces very rapidly. So in a good year, it may not matter that you're at the bottom of the plant or laid too many eggs. But in a bad year where it's senescing quickly, it can make a big difference.
Okay, and presumably the other host plant,
Pedicularis,
has more plant defense compounds, it's more heavily predated. There's some asymmetry there as well, right?
Yep. Yeah.
The CCs won't go on potentially else. So it seems to me that's a factor also. You might have some more unidirectional gene flow hybridization.
Yeah.
Host preference.
Yeah. Definitely.
I guess my last thought is you've got 5 different host plants in here, and the extent to which other host plants might provide a bridge in preferences between these things and how they.
Yeah.
So you just pulled out 2 host plants out of a complex system.
Yeah, Michael actually brought that up to me as well, and it turns out that what's different here is what's unique is Collinsia use. The other host plants are often used together.
Right.
So it's not clear there would be any sort of bridge to Collinsia.
Right. Okay.
But there are lots of bridges between the other hosts.
So what would happen if you added Plantago to your cafeteria choices of oviposition sites, just to see how they react to it?
Yeah.
They haven't experienced it, presumably.
Every once in a while we stick a foreign host in there, and in general they don't like it as well as their favorite host, but they may like it better than the other one. Yeah. There's a population on the east side of the Sierra near Schneider's Meadow, which Mike has also studied in detail, and that population switched from Collinsia to Plantago, and Plantago there was introduced. So they switched to this invasive plant very rapidly. What he observes is that host preference evolves very rapidly, and everything else is very slow thereafter. So within 10 years, you can see 100% preference for this to like 80% preference for that. But the physiology and the clutch sizes and the geotaxis hasn't changed.
Actually, that sort of answers another question I had, is the extent of genetic correlation between these traits. It's possible to take these through the F2 when you look at it.
Yeah. I'd like to do that.
Look at that.
The rearing is really difficult, and that's why we haven't done it yet. And now all of our hybrids are dead, so we'd have to start again with the F1s to get to the F2s.
Yeah. Are the F2s fully viable, or is there some outbreak compression?
As far as we can tell, yeah, but we haven't looked at it in detail.
Yeah.
We had all these hybrids laying in nature, and their eggs hatched, and their larvae did fine.
So you say when you have them in the hand, you can't tell them apart morphologically, but that you immediately can tell which is which.
If you put them on a plant. Sorry.
Well, from behaviour.
For
Well,
behavior.
for behavior.
It's so different that it's
Okay,
obvious.
so it's not something that's good on your hand.
Yeah, sorry.
Okay, any other questions? So if not, Lindsay can hang around for a bit longer for those who want to talk more about the research. But that was a really fine presentation. Thank you.
Thanks.